Hi, my name's Joe McKenna. I'm a research fellow at the University of Warwick and I'm a plant cell biologist, so I use microscopes to try and understand how plants are organised and work. So all the projects I've worked on have been involved in the organisation within the plant cell, so how the components within them, the organelles, are organised and how they interact with each other and they move around and things. What I'm working on at the moment is on the plant vacuole, and I'm trying to understand, so when you have a seed, the reason why seeds are good to eat is because they're very nutritionally rich. So when the developing embryo develops, obviously it can't photosynthesize light at first, so there's these specialized protein storage vacuoles, lots of them, like little bags filled with special proteins that are broken down. And we are trying to understand how these protein storage vacuoles are formed and how they are made. So my career pathway, I started my undergraduate degree at the University of Birmingham. I'm a bit unusual and I never had a burning desire when I started to go into plant science or anything. But I was through a very serendipitous event, I was really lucky. I undertook a third-year project with Professor Nonnie Franklin-Tong while I was at Birmingham, and it was looking at pollen tubes and different down the microscope, watching them grow and looking at different signals within them, and I fell in love. As soon as I looked down the microscope and saw these things growing, you watch them growing in front of you and streaming and all these things going on. I absolutely fell in love and knew that's what I wanted to do, and since then I've mainly used microscopes and cell biology to try and understand how things go on. I then moved down to Imperial, where I did a PhD, a master's and a PhD, and then I moved up to Oxford Brookes to work under two very big people in the field of plant cell biology, John Runyons and Chris Hawes, and that was a fantastic time, learning lots. And then I've recently, in 2021, I moved to Warwick University, where I work with Lorenzo Frigario on protein storage vacuole formation. So the most rewarding thing, without a doubt, is when you have that aha moment. You have some results that don't make sense, something's not happening, and then all of a sudden you see something or something clicks and you understand, you find the missing piece of the jigsaw, and it shuffles in your mind and it all makes sense and it all becomes logical. The first time so we do a lot of cloning with fluorescent proteins, and the first time you look at something down a microscope and you see where it's localized to, you see where it's gone to, and sometimes you might be the first person in the world to have seen where that particular protein is or that particular process is going on, and that's just phenomenal, and you're watching it live in front of you happen, and that's brilliant.